Okay, looks like we are at high noon Eastern time here. So my name is Andy Santacroce. Welcome everybody for joining our session today. Um, my role is that I am currently the president of the Eberly College of Science Alumni Society Board, and been introducing each of these sessions as we move along. So happy today to introduce Andrew Reed. Um, I will just give you some background here on Andrew. So again, welcome Andrew. Andrew is a evolutionary microbiologist originally from New Zealand. He did a Doctor of Philosophy in evolutionary biology at the University of Oxford. He has held various positions and fellowships at Oxford and then at the University of Edinburgh in Scotland, where he became chair of natural history. He's taught ecology, evolution, microbiology, and statistics. He has authored more than 250 peer-reviewed publications, 30 book chapters, and 4 edited volumes, and has been elected to fellowships from the Royal Society of Edinburgh, the Institute of Advanced Studies Berlin, the American Association for the Advancement of Science, the American Academy of Microbiology, the Royal Society, and the American Academy of Arts and Sciences. In 2007, Andrew moved to the Center for Infectious Disease Dynamics, Penn State University, where he is Evan Pugh University Professor in Biology and Entomology, Eberly Professor in Biotechnology, and Director of Penn State's Huck Institutes for Life Sciences. So thrilled to have Andrew with us today for our session. And with that, Andrew, I'll turn it over to one last thing before I turn it over to you. To all of our participants, at the bottom of your screen, you will see that there's a chat function. I see a few of you, saying hello in there, but I would like to direct everybody to the Q&A portion. So for any questions that come up along the way, I will continue to monitor that and jump in, with your questions. So please use the Q&A function to submit your questions, and that way we can keep track of them, make sure that they get answered, or as we move through, if any don't get answered, that we'll be able to follow up with those afterwards. Okay, so with that, Andrew, thank you again for joining us, and over to you.
Thanks very much, Andy. Really appreciate that. Wonder if I could just say at the outset, as you'll see as my talk goes on, there are various messaging issues that become quite serious. And so I'd be really appreciative of anybody who's either in the media listening or who is planning some wild and crazy social media outreach, if they could maybe contact me first so we could just agree the messaging. So I'm going to talk about how viruses change when they invade new hosts and what happens when they are evolving in vaccinated hosts. I've been working in this space now for 25 years, and I used to have to explain in great detail why this was important, and I no longer have to do that. Delta variant, I think, alone is showing the importance of pathogen evolution. What I'm going to do is talk about 2 examples of my former work, which I think is highly relevant to the current pandemic situation. And then I'm going to move to talking about the virus itself, the pandemic virus itself. SARS-CoV-2 is really unique in that it's the first time we've had in humanity a disease spillover which has gone pandemic at the same time as we have swung into action with vaccination. So there are 2 very novel things going on here. We've had spillovers before, HIV, for example, other coronaviruses. But we haven't had it on this scale, obviously, and we certainly haven't had with vaccination around straight away. So I'm going to talk first about a situation in rabbits where we've been following for about 70 years now a virus spillover event. Then I'm going to talk about what's happening in a vaccinated situation in agriculture. And then I'll try to tie that together with what I think is going on with SARS-2 now. So let me start with what in my field is one of the most famous cases of spillover events. So Europeans, when they got to Australia, missed rabbits and they wanted a rabbit industry. So they introduced rabbits and they certainly got themselves a rabbit industry. but they also got themselves that total devastation on an ecological level as the rabbits bred like, well, rabbits. And so they started working on biocontrol. And in 1950, a virus called myxoma virus, which is a pox virus, which they were experimenting on in field enclosures, jumped into the wild population and spread throughout the southeastern part of Australia. And it absolutely devastated the rabbit population. Within 6 months, the virus had spread over 1 million square miles, and at least tens of millions of rabbits, perhaps hundreds of millions of rabbits, died in those first 6 months. This is the most devastating disease outbreak in a vertebrate ever recorded, and absolutely totaled the rabbit population. And this is an example of the sort of devastation it had. So that's the MYXV, is the myxomatosis virus, and it basically reduced the rabbits to almost nothing. And over the subsequent decades, the rabbits came back to some extent, and the Australians introduced another virus, rabbit hemorrhagic disease virus, which had similar impact. But myxomatosis itself saved the Australian economy in the order of $1 billion Australian dollars. So that's by far the most cost-effective intervention, maybe in all of agriculture, but certainly with involving biocontrol. At the time that the spillover happened, Frank Fenner, who's one of Australia's finest scientists, recognized that this very lethal virus had just spilled over into a population, and there was bound to be evolution of the virus and perhaps of the rabbits themselves. This is an incredibly insightful realization. And he straight away dropped everything else he'd been doing and pivoted to work on myxomatosis. And his brilliant insight, and this really is, I still think it's breathtaking in my field, was that he could, even this is before the era of genomes, and they didn't even know about genomics. They didn't even understand about DNA evolution and so forth at this point. But he recognized that if he took from rabbits in the field the virus and put them into rabbits in the laboratory, he would be able to track changes in the virus over time. Okay, so the virus he's expecting to evolve, and he needs to keep testing it in a standard condition, and that's the laboratory rabbit. And so he set out and did that in the 1950s, did extensive work on that, really fantastic work. His graduate students picked it up again in the 1970s. And as you're about to hear, we picked it up in the 1990s. Well, at least we did it recently. We picked it up with the 1990s viruses. But what Fenner first discovered was within a year of the spillover that there was evolution taking place, and that this highly lethal virus had become less lethal almost straight away. And so those 100 million rabbits or so died, but that was imposing very strong selection on the virus. So this is a picture of myxomatosis in a contemporary rabbit in Australia. You can see it causes all sorts of horrible lesions and things, and it kills the rabbits. Now, this is in COVID speak what we would call 3 variants. Okay, the green and the red and the blue are 3 separate variants. Taken from this data comes from those early days, and that is along the x-axis. There is the day after a deliberate infection in these laboratory rabbits. So starting at 6 days in, a week or so in of infection. And what Fenner and his colleagues did was to put the mosquitoes that transmit the virus onto the rabbits and look to see how many mosquitoes got infected. So that's a measure of transmission on the y-axis. What you can see is that the original strain, which killed almost all rabbits in 10 days, that's the green one, did pretty well on infecting mosquitoes, but obviously only up until 10 days or so when all the rabbits were dead. And after that, there's no further transmission. Then another variant, what I've called here the less virulent one, killed half of the rabbits in a month or 2. And that one, therefore, transmitted to more mosquitoes over that first month or so. And you can see over the first couple of weeks here that the less virulent strain is doing better because it's going to infect more mosquitoes because it hasn't killed its host. And then he found some benign strains, and you can see they don't actually transmit very well at all. So they don't do much damage to the rabbit, but they also don't get to concentrations, titers that allow transmission. So from these data, we can see that the original strain has a disadvantage in terms of transmission compared to this less virulent strain. And then these more benign strains also have a disadvantage. So in this situation, evolution should favor the less virulent variant, this red strain. And that's actually what happened. So this is the percentage of rabbits that were dying, the laboratory rabbits that were dying from the virus through time. So he takes the virus in 1950 and 1955 and so on, puts them into lab rabbits and sees how many of those died. And so immediately the virus evolved to kill fewer rabbits. Though I do want to emphasize that's dropping down to 70%. So 7 in 10 rabbits still die. So that's very, very lethal, but not as lethal as the initial virus. And then over the subsequent decades, it sort of seemed pretty stable. And then there was talk in some very small studies that were done in the 1990s that the virulence might be rising up again, that the case fatality rate might be rising. And when I moved to Penn State in 2007, got very interested in trying to analyze this because we were now in the era of genomics and we could actually take a look and say what actual evolution took place and where's it going and so forth. So we actually were able to excavate in the freezers of Fenner's labs, and Fenner died around the turn of the century, but we excavated out viruses from his labs, from his graduate student labs, and some of the ongoing work that was happening in Australia. And the we here is these collaborators here. The bottom right-hand corner is experimental work we're doing in the lab at Penn State. The top right corner, Peter Kerr, is the one who made this possible. He was a grandchild of Fenner, if you like, and he's the last myxomatosis virologist still working extensively on this in Australia. And that's the phylogeny that we did of the viral strains that we got from the Fenner freezers and so forth. So at the top left is where the ancestor started, and then you can see the tree of life as the thing is radiated out. The ones in reds are the ones we did experiments on. The question we started with was, well, what are the 1990s viruses like? And this is just one example. So on the left is the variant that was released into the wild. And on the right is the variant, one of the variants that we worked on that was isolated in 1993. So we put this into lab rabbits, and you can see that the original progenitor, it kills everything in a couple of weeks. This one we isolated was isolated by others, but we used from 1993, actually killed everything faster than that. So it's actually become more virulent. So more virulent than the ancestor. And when we started looking at what was going on the left, I've got some pictures of the growth pathology. You can see that the top left picture is the normal sort of lesion you get when the virus is starting the infection. There's none of that in these 1990 strains. So you don't actually see anything that looks like classic myxomatosis. Instead, you see hemorrhage, a lot of hemorrhage, edemas and so forth. And then an awful lot of bacteria going in an awful lot of organs. And what's happened is when we first saw this, we found it very scary. We thought some sort of bacteria had got loose. But in fact, these 1990 viruses immunosuppress the rabbits. So they turn the rabbit immune system off. And so their symbiotic bacteria go everywhere. And that's what they die from. So they don't have an inflammatory response to control their own bacteria. They have their entire immune system switched off by these viruses. And to me, this was quite a shock. The virus has now become exceptionally immunosuppressive. So in terms of this overall picture, in fact, on this graph, we can't even scale it because everything dies, but it dies faster than the ancestor. So it's actually turned around and gone up. We've now shown that with European viruses. The genes responsible, we estimate, arose sometime around 1980 or so. And this virus has got hotter and hotter through time. And so it went down to start with, and then it come back up, and it's gone higher than the ancestor. And I don't have time to go through the data, but if you look at what's happened, the reverse of the Fenner experiments, where you take a standard laboratory virus and you stick it into rabbits from the different time periods, shows that the rabbits have got more resistant. So if you imagine what happened, we kill off 100 million rabbits. The survivors are going to be the ones that were either lucky or resistant to the virus. And then you get the evolution of resistance in the rabbits. And so we've got the evolution of resistance going on in the rabbits and the evolution of virulence in the virus. And so what we've got is an arms race. It's just like, the Soviets come up with a new weapon system, the Americans come up with one to match. It's just like that sort of thing. And this arms race has cranked things up to the point where the virus is now more lethal than the ancestor if you put it in laboratory rabbits. If you, the virus in the natural rabbit, then it looks much the same as the 1950s. So the arms race is like they're both pedaling furiously and staying in the same place. And to me, that's quite an eye-opener, that the thing that we thought had basically stabilized has in fact gone into reverse and is now even more lethal. And this, it turns out to be true for the very few other viruses that have been studied in these sort of situations. So there's the myxomatosis one in both Australia and Europe, but the rabbit hemorrhagic disease virus, not as well studied, but the same thing has happened there. Virulence has gone up and the rabbits have become more resistant, and there's an arms race. West Nile virus in the States, in house sparrows, has the same arms race. And then house finches have a bacteria that originally came from chickens, and both on the East Coast and the West Coast, the arms race is ongoing. So these arms races always escalate. So the viruses are spilling over into new hosts that have never seen them before. If that gets going, then we seem to have an escalating arms race. And I don't know of any examples at all where we had a spillover event where there hasn't been an arms race that's gone for more and more virulence and more resistance. That is slightly difficult to reconcile with some of the natural situations where we do see viruses that are pretty benign, but the implication is that they are benign only because their hosts are resistant, and that if they were put in other situations or the ancestral hosts, they would be pretty lethal. And just to say, before I leave myxoma, that this evolution is actually escalating. So lineage C is something that we've just discovered. That is made up of lineages that were from the 2010, 2013, 2015, that sort of era. There are viruses in lineage B of that sort of age, but lineage C seems to have got onto a trick that's allowing the evolutionary rate to speed up. So the branch length there is the length of the branches on this tree are a measure of how much genetic change is happening. And it's been pretty stable right through to 2000. And then something in this lineage C has given it an advantage, and that rate of evolution is speeding up in that lineage. We don't know what that is, but you might think of that as something like the Delta variant got a trick that made it very, very transmissible. And then the thing is just off and running. So the lessons that I draw from that in terms of the current pandemic is that you always have evolution after a spillover, unless of course the spillover fails. The most obvious example of a failed spillover from the virus's point of view is SARS-1. So humanity was able to get on top of SARS-1 and eradicate it before it started to evolve. That's also been true of some of the Ebola spillovers as well. But if the spillover starts to run so that there's a lot, it's running in a species long enough to really be significant amounts of transmission, then you get adaptation. And what that means is that it gets better at the new host. So it drops in out of some random bit of parameter space into a particular zone, and then it starts getting better at the new host. And that's true in the laboratory as well. And here we mean getting better means getting more infectious, getting more transmissible through time. And so what we've had so far with SARS, with the alpha and the beta and the delta variants, is the adaptation of the virus to be more and more infectious. And that is the nature of evolution. You always get evolution for what we would call more and more evolutionary fit variants. And that's what we've seen in the field, and that's what we're seeing with SARS-CoV-2 now. And the lesson we draw from the wildlife ones is that this process can go on for decades. And in the case of myxomatosis, where it's pretty well studied, there's no sign of a slowdown. In fact, if anything, there's a sign of a speeding up. And I just want to say too, that in the cases where we've got the evolution, co-evolution of the host back again, then we get this increasing virulence. And in all the studied cases, that escalates. I should say that there's other cases I showed you are not as well studied as the myxomatosis case. That still remains the canonical best studied case in terms of the numbers of strains and the amount of experimentation that's been going on. Okay, so that's evolution after spillover. Now I want to talk about evolution in vaccinated populations. This is a timeline from the introduction of vaccines, antibiotics, and antimalarials into widespread use. So if you look at the antimicrobial drugs, the antimalarial drugs, and the antibiotics, they go into use. So you can see, for example, penicillin is this one here, and then goes into use in the Second World War. And then drug resistance is seen in British hospitals just a few years later. It's not that the penicillin stopped working, but resistance is first seen very shortly afterwards. And if you go down here, you can see some drugs where the resistance is popping up straight away. In fact, sometimes it even pops up in clinical trials. The antimalarials are the similar story. You bring in a drug like chloroquine, and then a decade, in the case of chloroquine, a decade or so later, you see chloroquine resistance in the field. The drugs don't last long. But the vaccines, these things that stimulate our immune responses, they go into widespread use, and then sometimes they can even eradicate the species. Like smallpox failed to evolve around vaccine-induced immunity and was eradicated. And then some of these other diseases, the vaccines have worked for decades and decades and have not been undermined by the evolution of the virus or the bacteria. There are a few cases. This is whooping cough. This is a pneumococcal disease responsible for some types of pneummonia, and this is hepatitis B virus. These vaccines, the pneumococcal vaccine had to be tweaked to deal with the problem. The hepatitis B vaccine still works fine. These variants do pop up. They don't seem to transmit very well, and so the vaccine's still working pretty well. Pertussis, the vaccines are still working extremely well, although there is some ongoing evolution which might need to lead to changes soon. My point here is that there's a big difference between vaccines and drugs, and vaccine-driven evolution is quite rare, at least amongst the human diseases, and much, much rarer than drug resistance. And we've worked extensively on why this difference exists, but I don't have time to go into that today. I just want to say that I got really interested around the turn of this century in why it is that there are some exceptions. And I got particularly interested in this exception, which is Mareks disease virus, which is a really interesting virus in chickens. So it's a herpes virus closely related to the ones that cause herpes in humans. It exists in these sorts of chicken farming situations. It's a large DNA virus, so it has about 140 genes, so quite a lot bigger than SARS-CoV-2, and it's a DNA virus. The way it works is that if you shake an infected chicken, the virus literally falls out in the dander from the feathers, and that is breathed in, and that's how the transmission works. And it's a cancer-causing virus, so it causes tumors in the chickens. When it was first identified, at least the symptoms were first seen around 1920, but it wasn't really a big deal in the industry until about 1960, which coincides with the concentration of the industry and the commercialization of the poultry industry in these sorts of big settings. So this is a barn that I was visiting in Europe, but the American situations look the same. This is 30,000 birds, the same age, the same immunological status, all in a small space. And so a virus that gets loose in a situation like that can have a ball. And that's why I found it so fascinating. The virus became seriously problematic to the industry in the 1960s. And so a vaccine was developed, went into widespread use in 1970. About 10 years later, that vaccine was not considered effective enough, and so was swapped out for a newer vaccine, a second generation vaccine, and then later on a third generation vaccine. And this third generation vaccine is the reason your chicken nuggets are so cheap. It has stopped the evolution, at least the vaccine escape, of this virus. But this is one of the very rare cases where vaccine-driven evolution caused the undermining of a vaccine. And that's why I was so interested in it, because it is an exception, not the rule. Okay, so I want to say now that this is Joe Rogan, who many of you will know, one of the commentators, the American commentators, talking about one of our papers. And this happened, what does it say there, August the 6th. And since then, 3 million people have seen our paper on Joe Rogan's show. And over 400,000 people have actually looked at the actual paper itself on the website. And it is now, of all time social media papers, it is number 130 out of 18.8 million. So Joe Rogan really gave us a boost. And this is the numbers of people who've looked at the paper since we published it in 2015. But around 40,000 views before COVID hit, and then slowly escalating. And this is the Joe Rogan effect. So now we have 400,000 people who've actually looked at this paper. And what I want to do is just explain a bit about what's in this paper, because this is one of the very few studies of what can happen in a vaccinated population when a virus is able to evolve.
Hey,
that's
Andrew,
what he picked up.
Andrew?
Yeah, Andy.
If I could, we do have a question that came in from Chuck Cook. Mm-hmm. I can read it here. It should also be in the Q&A if you have visibility there. But can you please comment on host lifespan and its contribution to host resistance? It seems that in hosts with shorter lifespans, that resistance might develop more quickly. With humans where the viruses have a decided evolutionary advantage in the short term.
Yeah, that's absolutely true. So we don't expect any human evolution on anything like useful or relevant timelines. So historically, that human evolution has been really important. so obviously we would, if SARS-CoV-2 stays around and is still impacting human health and well-being, in decades or longer, we could expect some change, but we're certainly not looking there at the moment. Yeah, so the resistance that we might think about in the SARS case is all going to come from vaccination. I do think we do see on the human genome a huge amount of impact of disease selection, particularly malaria, but that's certainly not relevant in the current circumstances. My point on the myxomatosis is more that we get rapid evolution by the virus, and that's ongoing. And so that is what's happening with us in SARS-2 now. And it is true that if we don't have any other solutions, then the virus will have a huge advantage in the longer run. Okay, so so these are the experiments, the sort of experiments we did with the chicken. So we here as a group I've been collaborating with at the Purbone Institute in the United Kingdom. Obviously, if you're working with animal pathogens, then you have to have, there's containment requirements. There's a couple of places in the States, 3 or 4, where this can be done. But I started this work before I moved to Penn State, and so kept the same collaboration up and running. What we did was to infect birds in those isolators on the left, and you infect the chicks when they're young, and then they, over the space of a couple of months, they turn into full-grown chickens. So you go from a situation where you have to keep them warm to a situation where you have to keep them ventilated to keep them cool, because they really changed dramatically over this time course. And so we did experiments where we put in infected and uninfected chickens and looked at the transmission. And in those isolators, you can also suck the dust through the filters. And so you can look for the virus in the filters using the sort of PCR approaches that are used in human testing for SARS. Later on, the EU regulations changed, so we had to do floor pens, and that was perhaps a more natural situation, more like regular chickens. Although they, in our experiments, of course, they're running at a lower density than the industry does. We also did experiments with 2 types of chicken. The cob broilers is kind of the workhorse of what we eat, the ones that produce the meat we eat in supermarkets. The Rhode Island Red is about as close to the ancestral version as is currently possible to get. Now, I'm not going to show you all the results. I just want to show you, I'm going to show you one cartoon, but I've learned that it's a danger when I show people cartoons that they don't think there's any real data here. And I want to just emphasize, there's a lot of data in this paper, and there's a lot of replication and a lot of different experiments. I'm just going to show you one of those in cartoon. So in this experiment, we took this particular strain, which we would now call a variant in COVID speak, 675A, which was isolated from the chicken farms in the States, or a chicken farm in the States in the 1990s. And then we experimentally infected 10 birds with that virus. And then we put those birds together with 10 birds that didn't have any virus or any vaccines. And we put those together in our isolators and let the situation play out. And in those situations, what happened was that all of the chicks that we infected were dead by day 10, and none of the sentinel birds got infected or died. So this particular strain, 675A, is so lethal that it kills all of the birds. And it kills them so fast that the birds never become infectious. And we know that because, well, we measured it, but also when we take the dust from the filters, we can see that there's no virus being produced in the air. So this virus is so lethal, it kills all these birds before it has a chance to transmit to other birds. Then we took the first generation vaccine, we vaccinated those chickens, and then we infected them again with the 675A. And in that case, what happened was all of the unvaccinated birds died. And they started dying after 4 weeks, and they were all dead a bit over a month and a half. And we again know, because we're measuring the virus in the air, that we know that those birds that were vaccinated, they shed virus into the air. So the vaccine kept them alive, and they then were able to shed the virus into the air, which the other birds picked up, the unvaccinated birds picked up, and that killed them. So this is the fundamental result of our paper, that vaccines, which aren't able to stop transmission, can keep variants going that are more lethal, that are too lethal to persist on their own. Okay, so just want to say that again, what these experiments show, and as I say, there's a number of them, that the vaccination here doesn't stop transmission, keeps the birds alive. It's very good at that, keeps the birds alive. And that means that viruses that are unable to persist normally, that can't spread because they're too lethal, are now able to spread. And just to re-emphasize what's going on here is, so there's two variants here, and this is like what I showed for myxomatosis before. This is a density of virus particles that are going to be shed from the feathers through time for the two strains. So the left strain is what we would call a benign variant, and the right strain, the red one, is the 675A, the nasty one. And so if you vaccinate with the first generation vaccine, then it reduces the amount of virus that's going to be shed from the chickens, okay, by 2 or 3, 100-fold or maybe 1,000-fold reduction in the amount of virus. But it doesn't stop the shedding. The shedding still goes on. So virus is still coming out of these chickens being passed on. In an unvaccinated bird with the very lethal strain, the virus starts to be shed, but then the birds die and there's not enough virus has been shed by that point to spread on. If you then vaccinate, that strain is able to persist in those chickens because the vaccine is protecting the chickens from death. And that virus is now able to produce transmission stages that are shed into the environment. So the vaccination has kept the birds alive, and it made it possible for hot strains to spread to other birds. And that was our finding that we reported, and I'm still very proud of that paper. I think it stood the test of time very well. There are quite a few, quite a bit of discussion that I wish some folk had read in that paper, but the abstract is pretty blunt about, if you've got a leaky vaccine transmission happening, then highly virulent strains can spread. Okay, so, and that's the point that Joe Rogan was making about SARS-CoV-2. So what does all of this mean for the pandemic virus? Well, the first thing is we know, of course, that the virus is evolving like crazy. So the top is the tree of life for the virus from the original strain in Wuhan through the different, and this picture is now a little bit out of date, maybe a month out of date. Mu's not on here, for example. But obviously you all know about the deltas and the alphas and the betas, and there's lots of discussion about whether we're going to run out of Greek letters. The virus is evolving like crazy. And this is exactly the process you expect when a virus jumps species and is a new host. It's getting better and better at humans, and it's trying different things all the time. So some of these things seem to have a transient advantage, and then something else has a better time. And that's what's going on here. This is adaptation to humanity. And then I've got on that plot down at the bottom left, that's the relative frequency of the different variants through time. And then Delta, of course, is swept over the world. And I don't know whereabouts you all are, but certainly in State College right now, all of the cases in the hospital are Delta. So Delta completely took over Center County in Pennsylvania sometime in the middle of July, late July. And so we did get some alphas and some betas, but they were short-lived. Now, the key issue that we've learned from Delta is that the mRNA vaccines are still really great at stopping people from getting sick and dying. And that effect seems to be very robust. So against all the variants, the vaccines are extremely good at protecting people from getting sick and from dying. But much to, I think, everybody's disappointment, that it's turned out that Delta, at least, and I think there's some pretty good evidence that Beta can do it as well, are pretty good at transmitting from vaccinated people. Now, it's important to be clear here what we mean by that. The data are still very, depending where you look in the world, still very good on the fact that these vaccines can stop infections from starting in the first place. And in my view, the best data on this is showing that 2 out of 3 infections are prevented from happening at all in people vaccinated with Moderna or Pfizer. But 1 out of 3 is getting infected. That person is substantially less likely to die, get sick and die, very, very much better, but they are likely to transmit. And the surprising thing to me, at least, is that those people are as infectious initially as was somebody who's uninfected. So the difference kicks in how fast the virus is cleared. So the infectious period is shortened by the vaccination, but it's not prevented. I just want to make that clear again, that what we mean here is that people are not getting sick and dying at nearly the same rate. So it's a great thing to be vaccinated. 2 out of 3 infections are prevented. So that's a huge reduction in transmission. That's a huge impact in a population, much less transmission going on in a vaccinated population than unvaccinated population. But nevertheless, there is some going on. And in 1/3 of people, and the best data are actually from some basketball players that were being monitored in college athletics, which shows that the vaccinated players were as infectious. If they got infected, they were as infectious at the start, but they cleared faster. And so we've got vaccines that are imperfect, and so they're leaking. And that's what Joe Rogan's point was, and that's why he was so interested. And 400,000 other people are so interested in this work we did in chickens.
Hey, Andrew,
Yeah.
we have another question for you. This one from Stuart Lesson. Can post-vaccine viral infections continue in a vaccinated host, or does each infection boost immunity to post-vaccine infection?
Yeah, I think that's a really good question. We don't, I think, have terrific data on that. We actually, believe it or not, have better data on if you get infected first, then get vaccinated. We don't have terrific data that I've seen, at least, on what happens in a second infection to somebody who got a previous infection. The data on the potential to get infected, so getting infected at all, shows that natural infection is better than a vaccine. So the 2 out of 3 that I've got here, it's more like 4 out of 5 or 4.5 out of 5 for natural infection. So that suggests that the transmission blocking effect is more potent for natural immunity than for vaccine-induced immunity, but we don't know that for sure. It's, to me, one of the big oversights in the current data available. Okay, so what do we make of all this? Because nobody's really interested in what SARS has done over the last 18 months. They're all interested in where it goes next. And in light of the work that we've done, I want to offer the following speculations. However, I do want to emphasize that evolutionary biology, my profession, we are really good at explaining what happened. We're like historians, and I think we're actually pretty good at advanced hindsight. We're really lousy at prediction. And the reason we're lousy at prediction is it's much worse than weather forecasting. It's not just physics here. We've got all sorts of problems with trying to imagine what sort of phenotypes are out there that the virus can get to before they've arisen. And we just don't know before something's happened whether it's possible or not. And then there's all the issues about, rates of recombination, waiting for the right mutations and so forth. But to me, the main problem with prediction is if you tell me what phenotypes are possible and how they are related to infectiousness, I'll tell you what will happen. But nobody knows what's possible. And that is a really serious limitation. Like with the myxomatosis, we never knew it was possible for that virus to switch off rabbit immune systems the way it's evolved to do. Nobody in the 1950s could have predicted that, but it's got a gene, the virus has got a gene which is really good at turning off host responses. And that can only persist in a resistant rabbit population. It's just too nasty otherwise. Okay, so we got the Wuhan, original Wuhan outbreak starts off. It drops into humanity. It's got some level of transmissibility, and it's got some level of disease severity associated with it. And this is, just pretty random. Okay, so SARS-1 was less infectious and caused more severe disease. So SARS-1 is up here somewhere. And then we've got the alpha and delta evolution. We absolutely know that alpha and delta are more transmissible from both vaccinated and unvaccinated people. So they are adaptations to humanity. Even if there were no vaccines, alpha and delta would have spread like wildfire because they have very significant transmission advantage. Alpha is about 50% more transmissible than the ancestor, and delta maybe 50% again on top of that. So this evolution we've seen so far is all tied up with this adaptation to humans and getting more and more infectious. There must be a limit to that. We can't have a virus that's going to be instantly infectious forever. So there must be some constraints on what's possible. We just don't know what constraints are possible. That's sort of what we do there as a counterfactual is imagine the Darwinian demon, something that can just spread instantly to everybody. And there are constraints. It can't be like that. But we just don't know whether delta is at the constraint boundary or still miles away from it. My best bet is it's not at the constraint boundary yet. But if delta remains the dominant variant for the next 5 years, then we'll be able to say it's a constraint variant. their constraint boundary. There has probably been some walking up on this disease severity scale. So it's very hard to actually work out the disease severity for a variant in a human population. We can't do the sort of experiments we could do with rabbits or for chickens. The evidence is either that Delta is more, if you get it, you're sicker, or it's about the same. It depends on the study, but it's certainly much more infectious. And then going forward, I think what we're likely to see, especially only in rich countries where we have high levels of vaccination, we are likely to see the evolution of vaccine adapted variants. And that's to say ones that have a transmission advantage in vaccinated hosts. And that we will see as declining vaccine efficacy. So like we do with Delta, the vaccines are not as good against Delta as they were against Alpha or Wuhan. And I think we'll see more of that. But I want to emphasize that the Delta evolution at the moment, it ripped through India in the absence of vaccination. So Delta is just a general variant that's better at everything, vaccinated and unvaccinated. And I think we're going to get some new variants in due course, which are specialists on the vaccinated. And then the question is, what's going to happen to disease severity? So that's the disease severity in an unvaccinated person. Remember, disease severity is reduced in a vaccinated person. The vaccines are designed to keep us healthier, and that's what they do. So the question is, what will the variants that evolve in the future do to the unvaccinated? And we could get more severe variants appearing, like we saw in the Mareks case, or we could get a decline like we saw initially in the myxomatosis case. Remember, the variant was the one that was released in the myxomatosis case is so virulent, it was better off to kill at a lower rate, and therefore it spread. And so, this is what we saw in the myxomatosis case, at least initially. I've mentioned pertussis in both these cases. Whooping cough have some variants which are producing toxins at a higher rate, so do more damage, and then some variants that have lost some of their proteins, and so they function less well. So pertussis probably on a whole has come out somewhere in the middle, but particular variants are more or less severe. And then flu is probably a march every year, at least within a particular H and N combination, a march through different antigen space without much change in disease severity. And so we've got those three options at the moment. And to me, pretty much any of those things is possible. And I could sit in my armchair and produce predictions about any one of those. If we actually knew how disease severity affected transmission, then I think I could actually make some predictions. We expect that the more transmissible variants are going to spread. And so if disease severity reduces spread, for example, if people get isolated in a hospital or quarantined at home or whatever, then it could be that keeping people healthier with the vaccines will increase the spread of those. On the converse, it could be that variants that have an ability to get around vaccinated people also have a change in the spike protein, which makes it less effective at binding to host cells. So it's got immune escape, but it's less good at the non-immune. And so virulence would go down. So at the moment, we really, I think we've got the principles sorted out, but we're really stuck on it. But just let me say a couple of more words about what happened with the chicken virus. So this is Pennsylvania. This is a farm not very far from State College where we did a lot of work. This is inside that barn. This haze is the chicken dust. That's trillions of copies of Mareks disease virus in the air. This is the virus through time in that year and a half or 2 years in that barn, in that facility. And the virus is there all the time, but the chicken industry is doing just fine. So the first generation vaccines are not stopping transmission. You can see it's continuing, all that's persisting. But the farmer is happy enough with the productivity. So we can coexist with this sort of situation, and they do. However, they do get outbreaks sometimes, and then they use the second or third generation vaccines, and the third generation vaccine itself has never been beaten. It's just a bit more expensive, and so it's not the default position. But if they do have an outbreak, that's what they use. And they use the third generation one on the birds that lay the eggs because those are more valuable. And Mareks disease itself, the evolution caused a huge rise in the number of birds lost to the disease. Vaccination nailed it, the first generation vaccine. The rate of improvement slowed down, so they brought in the second generation vaccine that further reduced things, and now the third generation vaccine has made it more or less a non-event in the industry. So better vaccines were the answer in the chickens. And so to me, the lesson we might take is if the situation played out like it does in chickens, we're going to need to improve the vaccines. But we should recognize that these are life-saving interventions that have the potential to change the evolutionary landscape most likely they won't, because they didn't in other diseases. If they do in this, it could be good or bad. And if it's bad, we need to start tweaking with the vaccines. A year ago, Dave Kennedy and I, as a professor in biology, wrote the piece on the left, where we were actually reading it now. It's kind of interesting, sort of hypothetically interested in the possibility that virus evolution might start to take over. It was before the betas and the deltas and alphas and so forth. Then since Joe Rogan, I felt the need to, I've had trouble getting the messaging correct in the media. So I wrote my own piece there. And I would urge anybody who's interested in anything that I've said that wants an overview of this picture to have a look at both of these articles. In particular, the last one, the one on the right, is dealing with this question of why I still think that, although Joe Rogan was advocating very serious concern, and the implication of what he said was that, and many people in the Twitter sphere, that we should give up these vaccines because of the possibility, the hypothetical possibility of evolution. I'm very strongly negative on that. We've got life-saving vaccines that are demonstrably doing good. The possibility of evolution in the future is something we definitely need to worry about and think about, but it is not a reason to withhold these life-saving vaccines now. And with that, Andy, I think I'll stop and take questions.
Thank you, Andrew. And we actually, just very timely, we just had another question come in. So first off, Eric, looks like Eric Bilski is who this question is from. Thank you, Dr. Reed. PCR-based diagnostic tests seem to target stable genes and regions of the RNA genome of SARS-CoV-2, as I think the current assays work for the Delta variant and previous variants. Can you please comment on this?
Yeah, it's a really interesting question. We're doing a lot of wastewater surveillance at the moment, so we're very interested in whether or not there's any evolution at the PCR targets. So not in SARS yet, but in other situations, we have seen the evolution of diagnostic resistance, where the pathogen has evolved to become invisible to the tests. And that would be and you can see why. If you get tested and then isolated, the transmission stops. And so if something a person would be misdiagnosed as being not infectious and in fact is walking around infecting people, that would be a terrific advantage to the virus. Yeah, that has not been seen yet in COVID, but it's definitely something to watch for. And we've looked for it in the sequences. We've been pulling whole genome sequences out of the wastewater, and we have not seen it yet, and nobody around the world's reported that. Some of the more recent tests often do several different targets in the virus, and so it's increasingly unlikely I think that's going to be a problem, but absolutely something to watch for, and would be expected if we kept doing the simple tests that the CDC was recommending, over a year ago.
Thank you. And at the moment, we have no open questions. If anybody does have a question and wants to pop it into the Q&A, we'll just take this opportunity, in case nobody does have any questions, to say thank you again, Andrew, for oh, we got another question. That usually does it, right? So another one here from Chuck Cook. If the virus's tendency is to become more virulent over time, why didn't the influenza wipe out humanity in 1918? There were no vaccines then.
I think the flu situation is extraordinarily interesting, and also important to recognize that the dynamics of flu evolution is driven entirely by the immunity of the host populations. In fact, flu vaccines don't have any impact because we never get seasonal flu vaccination coverage high enough in a season to impact the evolution of the virus. So it's being driven entirely by natural immunity. And what we know from flu is that you get changes in the H's and the N's, so that you get variants that are not seen by natural immunity as well. And so you get the succession of variants are changing. And after enough of those, people have seen enough of that they epidemics or the pandemics can die out. So that's natural exposure, and that could happen here. So not going to rule that out as an option here. The other coronaviruses in humans don't work like that. but this one might. So that would mean, for example, that you'd be a change in the spike protein that would give them an advantage in vaccinated or in naturally immune people. And then a little bit later, there'd be another change in the spike. Eventually, to run out of spike protein changes that are possible, and the thing would die out, and it would persist only in children who hadn't seen it before or hadn't been vaccinated. And that some of my colleagues here think that's the most likely route. And certainly that's a totally plausible route. I just think there are other routes as well. And in my, flu is a very important situation, but it is only one of the cases where we've had virus evolution in response to, new hosts and vaccines.
And I do see one additional question, two questions. So the first one, how do we counter folks like Joe Rogan who only take part of your findings to forward theirs?
Yeah, I think that's a question that actually probably would be better aimed at, folks in the College of Liberal Arts or folks in the communications, arts and sciences world. I have enough trouble with virus behavior. Trying to understand human behavior is really tricky. But let me say, I do think that we've got ourselves into and I don't think this is just true in America. Globally, we've got ourselves into a very tricky space where people like me can't talk about the science of evolution and of vaccination in any way without thinking continuously about the skeptics looking over the shoulder. And so I actually think that Joe Rogan, in many ways, did us a service of, forcing this issue to the forefront, at least in my world. And it's an issue we should consider. And it's not to say, and that's what I argued in the conversation piece, it's not to say we should stop vaccination now, but we should be looking for it. And most of my colleagues who are busy sequencing genomes are not looking for a situation like we saw in the Mareks disease case. Now, it might be that Mareks is really special, and I'm completely up for that possibility. But we don't know whether the similarities between the virus and the vaccine in the two cases is more important than the differences or vice versa. So we do need to have this dialogue. So obviously there are plenty of people writing to me every day about how yesterday I got somebody who was claiming that it was me single-handedly responsible for providing the science that would allow all the poor people in the world to be wiped out. And the argument being that poor people won't get the vaccines, and this particularly across nations, that I was solely responsible for all of that. So there are a lot of very serious communication issues out there. On the other hand, I do think we need to have grown-up discussions, certainly in the scientific community and public health community, about where this virus might go, how to keep an eye out for it, and what we might do to mitigate it. And I'd like to think that if you read the chicken papers that I've done, we have solutions in there to how to stop that. Most obviously by making sure the vaccines knock down transmission. At the moment, the problem is the Delta variant is so transmissible from vaccinated people.
Thank you. We actually have 3 questions in queue now, so we'll get to as many as we can before we get to the top of the hour. And I'll just take them in the order that they came in. First one here was Barbara Hurd. Do you feel that booster shots could affect viral evolution?
Yeah. Obviously, the data on booster shots are still really not very good. But I think to the extent that booster shots are likely to increase the host response, provide greater protection, that will cut down transmission some more. That probably will mean that there's less evolution going on in the vaccinated folk, and might head off some of the stuff that I've been talking about with the COVID, with the Mareks. So I'm a bit of a fan of that approach. Another one is to mix and match the vaccines a bit more. So if you've had Moderna, get Pfizer, or if you've had Pfizer, get J&J. But I'm not advocating any of those things. I just think from the point of view of what we want is a broader response, antibody response and T-cell response, and we want it to be more potent. And those sorts of things will do that. So too would natural infection, right? If you got vaccinated and then get exposed, that will be a massive boost. And that probably will mean that your ability to contribute to transmission going forward has gone down very substantially. But there are other reasons to get the protection up higher, right? Transmission is bad, independent of evolution.
Right. Okay, and I'm just going to read the last 2 questions, and then we'll close it from here. The last 2 questions are, number 1, would you please briefly comment on your opinion on the origin of SARS-CoV-2 virus? And the second question is, how strong is the research relevance on viruses between chickens and humans?
Yeah, so let me take the origins first. My opinion, and it's just an opinion, is that the most likely origin will transpire that it'll be a wildlife spillover, quite natural. And SARS-1 was like that, and it took several years to figure out the origin of SARS-1. And so the most likely, I think the most likely interpretation is that that's what's going on, and there are some pretty natural explanations for why it turned up in Wuhan, even though that's also where they have the Institute of Virology. But it's also not, it's not a probability of zero that it could have come from the lab. And we, in principle, with a lot of transparency, we could know that. If that virus was being worked on in the lab, that would be a really, really serious smoking gun. So far, we don't have that at all. So I'd say balance of probability is very likely to be natural. And I think the focus on the lab has been distracting. These SARS viruses are circulating in wildlife all the time. And we've got to be careful that we don't get another spillover. And, we've had several this century, right? We've had SARS and MERS and 2 minor ones, as well as SARS-2. And so focusing on the lab and not worrying about the natural spillover of these things seems, how many more? We've had 5 warnings now. How many more do we need? So we've got to, I think, get the wet markets under control. We've got to make sure we're not doing habitat destruction where bats are coming in contact with humans in different ways, all that sort of thing. So I think very, very significant that we need to start focusing on the wildlife side. And sorry, Andy, I've just forgotten the second experiment.
Sure. Yep. How strong is the research relevance on viruses between chickens and humans?
Yeah. So I debate that with myself almost every day and with my colleagues every second or third day. And we simply don't know. So, we've got these case studies, one of which is Mareks. The situation now in SARS-2 is actually not unlike what it was at the start of vaccination in the chicken. And so the variants that we work with took 20 years to appear in the chicken case. And I think if they popped up now in humans, it would take some time before we saw them, because if they pop up in people that are not sick, they'll by definition be hard to find. So they'd have to spread quite a bit before we'd notice them. On the other hand, it's a DNA virus and not an RNA virus. Chickens are not humans. We could debate whether we live at higher densities or lower densities in chickens, whether we have the right sort of immunity, et cetera, et cetera. So we just don't know. So I don't think in principle we can say it's irrelevant. I think that's too strong. If with luck, it'll turn out to be irrelevant, and the trajectories will take off in other directions. But in all of these cases, evolution can go in many ways.
Okay. All right. So with that, we're just about out of time. Again, huge thank you to you, Dr. Reed, and you can see in the chat, if you take a look over there, lots of thank yous in there. Other comments around, excellent discussion, very informative, and I think just very insightful, given how much we hear publicly, about how these things are evolving. But to really hear about the science behind it has been wonderful. So thank you. Thank you all of our participants for joining the call. We'll continue with these, so keep your eyes and ears peeled for the invite to our next one, and have a great rest of the day. Thank you
very much, everybody.
Mm-hmm.